Hello everyone, it's my great honor to introduce the HTIRDB, a resource containing a transcriptional atlas for 105 different tissues from each of 7 species of domestic herbivore. The HTIRDB comprises the self-generated transcriptomic data from 105 tissues from 2 female, domestic herbivores from 7 species, and an extra 28,710 related published datasets. To date, the HTIRDB is the most extensive transcriptome data resource that provides tools and functionalities gene expression study for herbivores. At the homepage of the HTIRDB, the species and tissue and samples panels link to information on the species and tissue samples. The sample information module displays the anatomical distribution and the tissue coding system as tables or diagrams. The HTIRDB offers 5 functionalities for data retrieval and visualization: electronic fluorescent pictographic EFP, expression, comparative transcriptomics, co-expression, and variation analysis. The HTIRDB provides a suite of 9 bioinformatic tools such as JBrowser, Blache and Pathway. Maloney. The HTIRDB also provides access to RNA-seq and gene expression values with the download panel and gives introduction and demonstration for using of the functions with the help panel. The data stored in the HTIRDB were obtained as follows. First, we generated data using transcriptome sequencing from between 100 and 105 tissue samples that were dissected from 2 healthy juvenile female individuals of cattle, donkey, goat, horse, rabbit, sika deer and 2 breeds of sheep. Second, we mined published RNA-seq of the 7 species that generated with Illumina from the NCBI SRA database. The gene expression data was stored in MySQL. Apache 2.4, PHP 7.4, and ThinkPH 5.0 were used to create user access to the content of the database. The web pages were constructed using HTML5, JavaScript Framework Vue.js, Plotly.js, D3.js, and Edgets. The EFP panel was built using Python imaging library build. The corresponding anatomical location of the expression of a gene is presented on a pictographic representation of each animal, with different colors. The users can request an EFP by typing the target gene PXN and the species simply, and the HTIRDB would generate EFP that demonstrates the differences in expression of PXN in different tissues of the exercise system, digestive system and respiratory and circulatory system in sheep and cattle. The expression panel, comprising 3 modules, provides a comprehensive platform to access expression data on mRNA or long non-coding RNA. The relative expression can be visualized as table and heat map. The panel comparative transcriptomics includes 2 modules that allow for comparison of gene expression. The expression of target genes can be compared in the same species between any 2 tissues or in 1 tissue between any 2 species. The co-expression panel creates co-expression networks by visualizing genes with similar expression patterns in all tissues, in any single species. The tool panel provides extra bioinformatics tools. JBrowser Viewer generates a graphical overview of the newly generated RNA-seq datasets. The Blache tool enables users to interrogate and download reference sequences. The Go UKGG enrichment function allows analysis gene function to explore the biological roles and pathways associated with target genes. In short, the HTIRDB is the most robust 7 species of herbivore and comprehensive 105 different tissues, and biggest 638 billion raw sequencing reads transcriptome atlas for herbivores to date. The HTIRDB provides quality assurance by optimizing the sample collection, data processing, and database construction. A diverse range of functional tools will suit users from multiple disciplines and backgrounds.